Hi, here I am yet again with another video for you, this time covering the remainder of topic 2.1, which is all about biodiversity. Right, let's start with a definition of the term biodiversity. You've got this on your note sheet anyway, so you can see there the number of species and the number of organisms within each species found in a region. Just to explain it with a couple of simple examples, here's some rainforest. Huge biodiversity, probably every single tree shown in this image is of a different species, and you've got loads of ferns and other things around as well. There's a coniferous forest, every tree is of the same species, so much higher biodiversity here because there's a much higher number of species present. As for factors affecting biodiversity, the obvious one is latitude. Very high biodiversity around the equator, drops off the further away from the equator you move, largely because around the equator you get a higher intensity of sunlight, not a higher amount of light in terms of hours, by the way. They of course will just get 12 hours of sunlight all year round, as opposed to somewhere up here getting 18 hours of sunlight in summer and 6 in winter. The actual amount of sunlight wouldn't vary, but the intensity would, and of course you get higher average temperatures as well. Right, second factor is predation. Predators are horrible things, of course. Look at that, and they go around killing loads of things, don't they, and wreak havoc on their ecosystems, vastly reducing biodiversity. Or do they? Right, classic case study involving the removal of predators involves Yellowstone National Park in the United States. In the 1920s, it was decided to kill all the wolves within the park. And you've got two images here showing the park before the wolves were killed off and then after the wolves were killed off. You can see a big difference. But of course, the interesting thing is it's this one. There's the wolf still present. That's where it's all luscious and diverse still. The wolf's gone. Look at it. Why is that the case? Well, as shown here, once the wolves were killed off, the elk, the big deer that live in the park, their populations began to rise. And what did they do? They overgrazed everything. The grasses, the woody species and everything suffered badly from overgrazing. So, maybe wolves aren't so bad. And after all, as we learned in the previous video, wolves are actually the same species as the domestic dog. So perhaps we shouldn't think of them so harshly after all. What about sharks? So they're mean, horrible things, go around killing loads of people every year and loads of other things as well. Surely we'd be better off without them? Well, first thing to state is that sharks do kill people. On average, a whole 5 people per year lose their lives to shark attacks. Compare that with 100 million sharks killed by people on average each year. Rather puts things in context. When we're comparing humans and sharks for nastiness, I think you can see where the finger should be pointed. Anyway, as shown here, populations of large sharks have fallen by 90% over the last 200 years. This has meant there's been an increase in populations of smaller sharks and rays. These smaller predators are then wreaking havoc on the mollusk populations and they're getting decimated. So again, the simple message is predators are good for biodiversity. Predators may be good, humans are generally not. Of course, say generally not. We can have some positive impacts at times, but largely, particularly agriculture, of course, is not good for biodiversity. We tend to grow monocultures of a single crop cornfield in the USA there, wheat field in this country here. Don't need me to tell you that biodiversity is very low there. Right, let's move on to measuring biodiversity. Now, it's all very well me showing you a few images where you can easily see with the naked eye that one is far more diverse than the other. But what if it's not so immediately apparent? What if you wanted to do an actual study comparing the biodiversity of two areas? How would you go about it? Let's take, for example, my beautiful lawn against Callum's lawn, which would be more biodiverse. Right, here's a quick view of my lawn. Right, if I want to assess the biodiversity of this, what do? Do I get down on my hands and knees and count the numbers of every single plant on the whole lawn? I would, of course, be willing to do that for my class if necessary. Luckily, it's not, however. Now, what I'd do is I'd put down a series of quadrats as shown here, minimum of 10 quadrats, so they'll get a representative sample of the lawn, and count the number of different plants within each quadrat. But where do I place the quadrats? Can I just throw them over my shoulder or something, or look for a nice diverse area to put them down? Of course I can't. That would be biased sampling. This is of course a practical that in an ideal world we'd have actually been doing in college around this time of year. Lovely weather for it as well, but hopefully we'll still get to do it at some point. What you would do is you would mark out an area of your lawn using 2 tape measures. Generally I'd lay one tape measure there, a 10-metre length of tape measure, a second one here, and therefore I'd sort of effectively be creating a grid on my lawn. Grid with 100 squares. I would place 10 quadrats within that square, but how do I decide where to place them? Well, I'd use the points of the tape measure to decide on coordinates. I'd have an x coordinate here and a y coordinate here, and I'd just use a random number table such as this one to decide on my coordinates. My first quadrat would go at 8, 0, second one at 9, 4, third at 2, 5, and so on. Then, as I say, within each quadrat you can either count the number of individual plants, or sometimes we just record percentage cover. However, being a dedicated scientist, in my case I counted the number of individual plants. Callum, you of course are also a dedicated scientist. I'm sure you would count the number of individual plants as well. Right, just been out for a couple of hours, really tired now, but I've done all my work, counted all the plants I could find in 10 different quadrats, and here's my totals. Right, can't actually claim to have gone to Callum's lawn and done the same thing. Can't unfortunately claim that Callum's gone out and done this and sent me these results either. I haven't actually asked him to. I'm sure he'd have been more than willing to do so had I asked him though. But I've guessed at Callum's results. Now, let's have a good look at this. Well, what would happen if these were the results? First thing that would happen is Callum might turn to me and go, ha ha, my lawn's more diverse than yours because I've got 6 species present and you've only got 5. What would I do in return? I'd give him a good clip round the ear, first of all, for being cheeky, and then I might point out a second thing. But wait a minute, young Callum. Your lawn might have more species than mine, but really, your lawn is virtually a monoculture of grass. You've got a tiny number of other things in there. Mine is really far more diverse than yours. How do we actually decide? We can't just look at the total number of species and let Callum win 6-5. Not going to let him beat me after all. How can we actually calculate which lawn is more diverse? Right, I'll illustrate this using some different results to begin with, because I want you to calculate the biodiversity of mine and Callum's lawns. Imagine that other lawns had been sampled. They hadn't even bothered with the grass this time, and they just got these results. Now, for sample 1, you can actually quote, or calculate, to say, something called a Simpson's Index of Diversity figure. To do so, sorry, I had to rotate the phone here. You might have to rotate your screen when watching this. You would lay the results out in a table like this. I've just laid out the results for area 1 only. I've written the number in the first column. I'll then put n minus 1. Little n is the number. N minus 1 is literally that, as you can see. I've then done n times n minus 1. So in the first row, it's just 300 times 299. The bottom, I've added up the total number of individual plants, and that is slightly confusingly given the notation of a capital N, which is 1,000. I also add up the total of the final column. That figure at the bottom there is the Greek letter sigma, which just stands for sum. I've just totaled up all of these, and that is the sum of n times n minus 1. To calculate the actual biodiversity, what's called the biodiversity index, I do the sigma figure at the bottom of the final column divided by the capital N times capital N minus 1, such as that, and I get this figure. The figure for diversity is always between 0 and 1, and the higher it is, the more diverse the area is. Right, let's go back to the figures for mine and Callum's lawns and set you a task. Can you calculate the index of biodiversity for both of these? Pause the video and draw out the tables. Come back when you're done. Right, here's the answers for my lawn. Hopefully you've understood the process and managed to do this. And you can see the final figure there, 0.569. Callum's rather boring monoculture of a lawn, 0.0599. So, in the end, it's not even close, is it? I win easily, in your face, Callum. If you want some sort of further guided examples of how to calculate the D figure, the index of diversity, sometimes you even see it abbreviated to S rather than D, S for Simpson's Index of Diversity, then I've sent you a link to a document which explains how to do this anyway. Right, everything I've covered up to now has really been about species biodiversity. And as we've learned, a habitat has a higher species biodiversity if there's a greater variety of species present. But there is a second type of biodiversity you need to learn about, and that is genetic biodiversity. In fact, I'll show you another image and then come back to this one. Right, yeah, this nice little image summarises the main difference. We already know what species biodiversity is, hopefully, but genetic biodiversity only really applies to a single species. You can either have a single population of one species with high genetic diversity, they look like that, or low genetic diversity if they all look the same. It's not, of course, just about appearance, it's about biochemistry, anatomy, physiology, etc. as well. But if they all look and function the same way, there is low genetic diversity. And of course, it's in a species' interest to have high genetic diversity. Right, let's quickly look at what causes genetic diversity. Go back to basics as first, as you have previously learnt, even at school, and also briefly with me when doing topic 2.2 before Christmas, that we have all our DNA is found on chromosomes, 46 chromosomes in our cells with 23 pairs strictly. Now, each chromosome contains a large number of genes. mutations, but actually the genes for a single feature are always found on the homologous chromosome. So as you can see from this one, the gene for eye colour would be located on 2 different chromosomes, and it would actually consist of 2 alleles. Now, on this pair of chromosomes, for example, there's not much, this is just for a single individual. Both alleles are the same for making earwax, but they've got different alleles for eye colour. And of course, for many features in organisms, there are a number of different alleles that could be present. These could be alleles for fur colour. You might have black fur, white fur. You might have alleles for tall or short plants. In peas, a classic one that you may have studied at school. Sometimes it's not only 2 possible alleles present. The human blood system has 3 possible alleles. There's A alleles, B alleles, and O alleles. Each individual can only have 2 of those alleles, but there are 3 alleles out there in the population. Now, a population or a species will have high genetic diversity if there are multiple alleles out there for one feature, and if there's a reasonable number of each allele. There's no point in having 2 alleles if 99.9% of the alleles for eye colour are blue and only 0.1% of them are brown. That is not high genetic diversity. If you've got a 50-50 mix, that's the highest possible diversity that you could get for the sort of eye colour allele. You'll have even more diversity if there was a 3rd possible allele in the population. So genetic diversity is sort of determined by the number of different alleles out there in the population and by the frequency of those different alleles. Now, high genetic diversity is a good thing for a species, basically, because it allows them to survive either changes to their environment or allows them to survive in a variety of different habitats. The classic case is, of course, this one of the peppered moth. I'm sure you've all studied this one before. You've got the normal light-coloured moth, very well camouflaged on trees in rural areas, but in a dirty, polluted place like Manchester, where it was first discovered, the black peppered moth on soot-coloured trees actually survives much better. So having these 2 different alleles for colour actually is a good thing for the peppered moth. If it only had the 1 possible allele, then there'd be no peppered moths in cities. You'd only find them in rural areas. So it allows them to exist in larger numbers. And what's more, again, predators are a good thing for this. Predators are not only good for maintaining species biodiversity, they're good for maintaining genetic biodiversity, because it's predators, of course, which eat the moths which stand out and the ones that aren't camouflaged. If there was no predation, then the black ones would have no advantage in the cities and the light-coloured ones would have no advantage in the countryside. Various other examples of genetic diversity in mice, actually in jaguars here, the normal colour one there, but you do get some black ones. Of course, humans show a reasonable amount of genetic diversity as well. Now, sometimes genetic diversity can cause problems. Sickle cell anemia is a genetic disease. Probably come across it before at school. If you have sickle cell anemia and all your red blood cells are shaped like that, that is a seriously debilitating disease. However, if you have sickle cell trait, which basically means you have 1 normal allele and 1 allele for sickle cell disease, then actually the majority of your blood cells are normal and you just have a small number of sickle cell ones. You might think, well, that's still a slight disadvantage. Is it? doesn't seem to hugely affect people, to be honest. It might put them at a very slight disadvantage. They have to perform high endurance events, but in normal life it wouldn't generally affect them. But it also gives them an advantage, again, showing that even seemingly harmful things can actually provide an advantage at times. People with sickle cell trait are actually pretty much, or certainly show far more resistance to malaria than people with normal red blood cells entirely, and therefore sickle cell allele is actually present in far higher frequencies in areas of the world where malaria is endemic. So again, as I say, even a seemingly harmful allele can have beneficial effects, and again shows the sort of beneficial effect of having genetic diversity in a population. Okay, and that's pretty much it for today. With that, you should now be able to go on and complete the topic 2.1 note sheet, complete sections D and E on the topic 2.1 question sheet. You can also go ahead and complete homework 21 at some point this week. And that's that's work to last you for the whole of this week, as mentioned on the email. But that's it for today. Bye now.